at the College of William & Mary, Virginia Institute of Marine Science on the spatio-temporal monitoring of nursery habitat effects on densities, survival, and growth of blue crab. His current research interests include spatio-temporal habitat modeling, ontogenetic habitat shift modeling, and the state space modeling of habitat effects on population dynamics. So thank you for your attention. Take it away.
Hey guys, thank you so much. Can everyone hear me? Good? All right. So thank you guys so much for letting me come up here and talk to you guys a little bit about some of what I did over the last 4 years. Yeah, my name is Challen. I just defended my PhD in July, so I'm what you would call a recovering grad student. I had a bad habit for about 4 years. I think I kicked it, but the doctor name still sounds a little bit weird for me. But my background is primarily at this intersection between quantitative or statistics and quantitative ecology. So I'm somewhere in between a statistician and a quantitative ecologist, and that's kind of what my work is, to use some of these more complicated and intricate models to get at some of these really complicated environmental and ecological processes. So I'm pleased to present some of my work today. Let me see, can I do this? Yes. So today I'm going to be going over a little bit about some blue crab ecology, some habitat stuff, some nursery habitat dynamics, and some ontogenetic modeling. So just as an outline for what I'll be talking about going forward, first and foremost, I'll give you guys an idea of the interplay between habitat and fisheries, some of what I did in some of my most recent work. Then moving into blue crab life cycle and basically blue crab ecology, primarily in the Chesapeake Bay, although I still think a lot of it applies down here in the Gulf of Mexico. Getting into a little bit about a crash course in nursery habitats and some of the characteristics that they employ. Going into some original motivating questions and thinking behind how I set up my PhD. And like most folks who go through grad school, the questions that you start out with don't necessarily become the questions that you end with. I see a lot of smirks from some of the more senior PhD students. So with that, we're also getting into some of the newer questions that I looked at in light of some of the changing evidence, because when you see things that conflict with your prior notions, you change how you attack the question into something that might be a little more representative of the dynamics of the system. And then finally getting into methodology, the results of this chapter, and some of the big conclusions and takeaways, which are some of the underlying themes of my dissertation. So and I'm going to talk a lot, and some of my stuff is probably going to be boring. You guys, your eyes might glaze over. I apologize in advance. If you guys ever get lost, Gerald over here on the bottom of your screen will be holding up cue cards, kind of giving you an idea of where am I in the introduction, am I in the methods, results, conclusions, takeaways. So if your eyes glaze over, no fear, look down, Gerald will guide your way home, right? So first and foremost, I'm going to talk a little bit, or start with a premise that I think a lot of us already know intuitively, particularly for this audience, and that is that we're losing important marine and estuarine habitats at an alarming rate. Now, this happens for a variety of reasons. First and foremost, for the Tampa Bay, nutrient pollution was something that was a huge impact for a number of years before you implemented a number of controls to reduce some of those pressures. But in addition to nutrient pollution, you have things like destructive fishing practices, so essentially taking bottom trawls and destroying hard bottom habitat. And then, of course, we have the big bad one, which is climate change. Essentially, thermal stress imposed on a lot of these foundation species like cold water loving seagrass meadows, coral reefs, etc. When you have that thermal stress imposed on those organisms will die and deteriorate, and the habitats that they encompass and produce will also decay and deteriorate. So we have a lot of problems. And when I'm trying to talk to people, specifically lay people, and I have them basically motivate an intention to care about some of these marine environments, the framework that I find most helpful, although I know that there's a number of different ways that you can make this argument, is through the lens of fisheries. People come to me all the time and they say, Challen, why should we care about seagrass meadows? Why should we care about salt marsh? And I say, you like to eat grouper? You like to eat flounder? Snapper? Essentially, drum? Pick a species of whitefish that you like that winds up on your dinner plate, and odds are it starts life in a seagrass meadow, a salt marsh, or some other structurally complex marine or estuarine habitat, right? And so we should care about that, because essentially when we impose negative stress on these different habitats, essentially that causes these habitats to decay and deteriorate. That impacts our fisheries. And first and foremost, that will affect our economic value because a lot of these fisheries are going to be bringing in, especially commercial fisheries, are going to be bringing in a lot of money. There's also going to be, particularly in the Gulf of Mexico, a large charter industry, which is also going to be relying on abundant distributions of fish. So if we lose those habitats, we lose that fisheries productivity, and that economic value goes away. But there's also a social implication, right? I love eating fish. I have like 16 pounds of whitefish in my fridge right now. I don't want to stop eating fish. And so one of the most motivating reasons for me personally to preserve a lot of these habitats is so that I can keep eating fish, right? And so there's both social and economic incentives for us to care about preserving these marine habitats. There's also a myriad of other ecosystem services that I'm sort of sweeping under the rug here. I'm only focusing on a motivation and a persuasive argument that I find is really motivating to a lay audience. And so when we talk about preserving and conserving and restoring these habitats, the framework in my mind works best like this. First, you illustrate the connection between the marine habitats, the fisheries that they produce and are responsible for maintaining, and the human system that is interplaying with those two dynamics, kind of just like I did in the previous slide. Then you work to identify all of the habitats that's used by a specific species throughout its lifespan. This is going to be an underlying theme for the rest of the talk. Then you identify the stressors that are influencing those different habitats that are important to that fishery species or that complex of species if you're looking at a community, for example. And so these are going to be things like thermal stress, nutrient pollution, destructive fishing practices, etc., right? And then finally, if you're lucky, you can work to reduce some of these stressors and maybe restore some of that habitat and then reap the benefits in terms of fisheries productivity. Again, a great example is the Tampa Bay 2009 seagrass recoveries, where after they reduced nutrient pollution, saw a rebound in seagrass meadow extent. Now, obviously, that doesn't always work. I mean, ask anyone trying to restore oyster reefs up in the Panhandle right now. But if we're lucky, we can begin to offset some of these negative consequences of our past behavior, and again, reap the benefits in terms of the fisheries that the fisheries productivity that those restoration events produce. And for the remainder of this talk, I'm going to be primarily focusing on this second bullet. I'm just one guy, and even though I spent 4 years thinking about all of these questions with respect to blue crabs, I am going to primarily be focusing on identifying the habitats that are used by these this species, at least as it pertains in the Chesapeake Bay, and how these animals are switching and when they're switching from different habitats as they grow and change throughout their life cycle. So that brings me to my study species, which is the blue crab, Challenectes sapidus. And I joke that if blue crabs were people, they would be the worst kind of people. They are mean, they are aggressive, but they also happen to be pretty tasty. And in addition, they are an economically important species, both within the Chesapeake Bay, where I just came from, and in the Gulf of Mexico. As a dominant benthic predator, they consume a number of smaller invertebrate species, things like amphipods, tiny itty-bitty crustaceans, different sorts of clams. In Chesapeake, that's something like Macoma baltica, the Baltic clam, or Maya arenaria, the softshell clam, as well as different sorts of polychaete worms, and then plant material, detritus, things of that nature. However, they're also an important prey species to a lot of things that we like to consume. Things down here like red drum, spotted sea trout, silver perch, southern flounder. There are a number of animals that like to consume blue crabs, including blue crabs themselves. These are a highly cannibalistic species. Like I said, they'd be the worst kind of people. And they're actually regulating their own populations quite robustly, especially up in the Chesapeake Bay. But there's one more arguably the most important predator of blue crabs. Can anyone take a guess as to what that might be? Humans. Exactly. The fishery, right? And specifically, I didn't look at the numbers for Gulf of Mexico, but at least for fishing mortality up in Chesapeake Bay, the predation pressure that we impose on this species dwarfs just about anything else that's out there. And so within the Chesapeake Bay, the landings for commercial landings for the blue crab are typically between $50 to $100 million a year, going back through the last 10 years. In the Gulf of Mexico, that number, looking at the NOAA landings data, was between 80 to 100 million. So obviously a good chunk of change. And because of these economic and these ecological incentives, we should care about the species and what happens to it, right? That's my motivating framework for why you should care about the rest of the talk, by the way. So in terms of the blue crab life cycle, we were talking with a few students before we got in here, and one of them mentioned, John mentioned the learning the Krebs cycle for was it your comprehensive exams?
Yeah, it was Krebs ecology book.
Yeah, so instead of the Krebs cycle, today you guys are going to learn the crab cycle, right? There's going to be one stage only, and the only stage is crab, so a little bit more simple, but hopefully you'll be able to get something out of it. Essentially, the blue crab life cycle begins with gravid females or egg-bearing females migrating to high salinity waters from the upper reaches of the estuary, where they will release their eggs. This orange mass underneath this female right here is essentially her egg mass. It's got about 3.2 million eggs. Now, afterwards, these larvae, or zoea, are going to be swept out onto the continental shelf, where they undergo a period of development for about 3 to 4 weeks and 7 to 8 different zoeal or larval stages. After that time on the continental shelf boarding school, they then reinvade the estuary as these bug-looking things up here known as megalopy. And this encompasses their larval phase of their life cycle. After they reinvade as these post-larvae, they rapidly settle as small juveniles, about the size, maybe twice the size of a pinhead. So these guys are itty-bitty, and they rapidly grow through a series of different juvenile stages that I'm going to refer to, from the purposes of this talk, as small, medium, and large juvenile crabs. Really complicated for you guys. And this is going to be the juvenile phase of this life cycle. Finally, these animals are going to molt to maturity and complete the life cycle of the blue crab. Now, a general theme in ecology is that the smallest and most vulnerable size classes of these animals are going to be the ones that are going to be most reliant on different habitats, specifically nursery habitats. So any sort of structure, highly turbid habitat, these are going to be the important areas, or these stages are going to be the most responsive to changes in nursery habitat and different estuarine and marine habitats. And so that's where I'm going to focus the rest of this talk. Now, folks may not know what a nursery habitat is, and so just in case that's a foreign term, broadly speaking, nursery habitats are habitats which disproportionately contribute more individuals per unit area than other candidate habitats. And they do this by having a few general characteristics in common. First, they have tons of places to hide. If you're looking at a seagrass meadow, for example, well, you're going to notice that they have lots of different interstitial places or interstitial spaces, lots of nooks and crannies for these organisms to hide in. And it essentially creates a barrier where predation efficiency is decreasing. They physically can't get into those spaces, right? But in addition, it's also just great refuge in terms of reducing visual predation efficiency, right? If they can't see you, the odds are you're not going to be as eaten at that high of a frequency. And so this sort of refuge capacity is a through line in terms of the dominant characteristic of a lot of different nursery habitats. In addition, the other characteristic is lots of food. These organisms are tiny, and they're going to need the resources to grow and develop. The faster you grow, the faster you outgrow the mouth of a lot of fish that want to eat you, and the better your chances of survival, right? And so when you're looking at, if you're going to be evaluating two different habitats to see which one might be a nursery and which one might not be, you're going to be looking at these traits. So in a seagrass meadow, you've got lots of places to hide, so that drum isn't going to be as lucky, and lots of moderate food availability. Now compare that with barren sand, which has fewer places to hide, so that drum is going to get lucky a lot more often. And also variable food. Sometimes sand flats can be absolutely unproductive. Other times they can have quite a bit of food availability in terms of bivalves, crustacea, and different sorts of polychaetes. But if these guys on balance were to give a Yelp review, Terry over here seems really partial to that seagrass meadow. And compare that to Reginald, who does not seem nearly as enthused about that sand habitat. So if you're a blue crab larva or post larva and you're going to be trying to find a new home in your new estuary, odds are you're going to be wanting to look at seagrass meadows a little bit more carefully than unstructured bottom sand. And this kind of gets into a paradigm for blue crabs. There's a reason I brought up seagrass so much. The dominant thinking for a long time was that juveniles settle into seagrass meadows until about 30 millimeters carapace width, or about 30 millimeters across. That's about the size of the tip of your thumb to your first knuckle for reference. Afterwards, they then would migrate to unstructured habitat. So the framework looks a little bit like this, right? They settle into seagrass, they stay there for a certain period of time, and then they migrate to somewhere else. Now, you guys might be thinking a natural question is, well, what's so special about 30 millimeters? Why is that number magical? Well, the thinking went that after 30 millimeters, these guys are bigger, they're more aggressive, and they have more prominent spines, so they're not quite as palatable. That drum won't be as lucky. And so what we call this is achieving a size refuge from predation. And because of that, you don't necessarily need the refuge that you relied upon in these different structurally complex habitats. That refuge characteristic isn't as important, and now you can exploit any habitat you want that may have food. There are some unstructured bottom habitats, particularly in areas where there's like the estuarine turbidity maximum, the place in the estuary where the water is cloudiest, that usually has really high food availability. And so these guys are going to migrate over there because, again, they don't necessarily need that refuge capacity anymore. Now, this is all well and good, but one of the things that we noted up in Chesapeake Bay was that we are running out of the dominant species of seagrass that has been the most important nursery, or thought to be the most important nursery for blue crabs in Chesapeake Bay for a long time. Zostera marina is a cold water species. It actually looks fairly similar to Thalassia. That was my first thought when I kind of migrated up there, because I was a seagrass guy down here under Tom, actually. And so this species really doesn't do well above 30 degrees C. And as those summer temperatures are getting hotter and hotter and hotter, well, you can just look at the projections from some of my colleagues at the Seagrass lab, or the SAV lab in the Virginia Institute of Marine Science. This was a publication from a friend of mine, a colleague of mine, Mark Hensel. It's out in, I believe, PNAS right now, but showing that under different scenarios, even under the unrealistic no more climate change scenario, we're still not really going to get Zostera to come back at even a smidgen of what it used to be back in the 90s, for example. There was looking like, as we reduced some of the nutrient pollution in the bay, that there was an uptick in Zostera marina cover up until around 2018. Some fresh water kind of squelched those gains. We had massive losses once again. And it looks like, because the summer temperatures are already at such a high level, that we're really not going to get any more bang for our buck, even if we include nutrient reduction measures and, the unrealistic no climate change measure. So we're running out of seagrass meadows, right? Rupia, for example, might be able to replace some of this, but we were thinking that it's probably a bad news story for blue crabs. And so the motivating question for me when I first came into VIMS, this is the context I wanted to set up, was, are there other habitats that might be able to offset some of these losses in Zostera marina, right? We know the seagrass is declining. We know that there are other structurally complex nursery habitats, oyster beds, riprap, salt marsh habitat. Maybe those might be able to compensate for some of the losses in productivity that we are expecting from that seagrass loss, right? Well, that was the driving question. So for my first chapter, this is out in, we got this out last year in Frontiers, but essentially we were looking at animals between 20 and 40 millimeters carapace width, and we were using survey data throughout the Virginia portion of the Chesapeake Bay, using the Virginia Institute of Marine Science Juvenile Fish and Blue Crab Trawl Survey. The reason we targeted this size class of animals is it's wrapping, it's port ending that magic 30 millimeter number, right? If these guys are emigrating from these nursery habitats at around 30 millimeters, well, if you target animals between 20 and 40, then hopefully you're getting the animals that are emigrating out of those nursery habitats, and it gives you an idea of how many animals those habitats are producing. But what we began to see is that as we were looking at our trawl survey data, you'll notice that there were some tiny crabs that we found adjacent to seagrass meadows, but the vast majority of them were up here. They were all in unstructured salt marsh meadows, particularly near the estuarine turbidity maximum. Now, this flummoxed me. I looked at that data, and that was my first thought. I was like, what is going on? Are my models incorrect? Is the data incorrect? What could it be? Well, we had a couple of alternative explanations. The first was that the difference in effective juvenile habitat. Basically, how much of these habitats are there? For example, if you look at these maps, and hopefully you can see at least the salt marsh. If you're wondering where the seagrass meadows are in that other figure, that's kind of the point. There's not that many of them. The overall area of salt marsh habitat in the bay absolutely dwarfs that of seagrass habitat, right? And so as a result, even if the per unit area productivity of seagrass meadows are higher than salt marsh, that signal might have just been swamped by the absolute magnitude of salt marsh relative to seagrass meadows. That was one hypothesis. Another was maybe some kind of logistic or sampling issues. Seagrasses tend to be in shallower water than the canals or channels adjacent to some of these salt marsh tidal creeks. So maybe it was just that they weren't sampling effectively in those areas. And the final one that I thought might be the most persuasive was, what if it's a dispersal issue? There was some evidence in literature that juveniles might be emigrating to other habitats before that 30 millimeter magic number. So if you're targeting animals after they've already moved, well, then it's no wonder that you're not finding animals in their original nursery habitat. And so that was the one that I wanted to focus on for the rest of my PhD. So to kind of give you an idea of what I'm talking about, essentially, I was thinking maybe the life cycle looked a little more like this, where these animals were settling into seagrass meadows, moving to salt marsh meadows after some size threshold, and then moving to these unstructured habitats later on in their juvenile and later adult life cycles. And again, if the trawl survey was targeting animals in this range after they've already moved, well, then again, it's no wonder that we missed the signal for seagrass meadows. Now, you may be thinking, well, what's the rationale for moving habitats? You know, some of these salt marshes are quite a bit farther away from seagrass meadows. Why would a crab risk all of that? And for that, it helps to understand exactly what the trade-offs are between these two habitats, right? So when you're looking at seagrass, this is generally characterized by high structure, lots of nooks and crannies to hide, and moderate food availability. Contrast that with salt marsh, which also has structure, but a lot wider interstitial spaces between these different marsh shoots. So moderate structural capacity and moderate refuge, but a lot higher food. It has a lot more polychaetes, a lot more crustacea, especially in these really turbid areas in the Chesapeake Bay. This might not apply more generally, so I want to check that at the door, but at least in my study system, this was the dynamic that was taking place. And so if you're a tiny blue crab, right, a lot of things want to eat you, and you don't necessarily need that much food. You're tiny, and so proportionally, you can probably get by with moderate food availability. And so generally, these guys are going to be prioritizing structural availability over growth, right? However, as they get larger, they're beginning to increasingly achieve that size refuge from predation that I talked about, and then they might be wanting to switch. You no longer need as much refuge. You still need some, but not as much, and you're going to be increasingly prioritizing food availability. For those of us in the ontogenetic business, this is sort of evaluating something called mortality to growth ratios. Basically, what are the driving concerns at the species at that given size class, right? And so, the first main question that I had was, how does juvenile blue crab nursery use change at different size classes? And the first thing that I wanted to look at was abundance at a per unit area level within some of these different faculties within the seascape. So abundance essentially provides circumstantial evidence for nursery habitat use. And here the hypothesis was that the smallest size fraction, about 15 millimeters across, it's about the size of your pinky nail, those guys are going to be in seagrass. Then these medium-sized animals between 16 to 30 millimeters, again, pinky nail size to about the width of your tip of your thumb to your first knuckle, those guys we were hypothesizing they would be high, their abundances would be highest in salt marsh, and then the largest size fraction of juveniles we considered would be highest in unstructured bottom. And this kind of gets at that revised life cycle that we were thinking based on some of this evidence from my first chapter. Now, but it doesn't really do enough to just look at abundance, right? The first thing you notice is that abundance could be purely correlative. There's a number of different reasons why you might have more crabs in one habitat than another, and it might have nothing to do with how attractive that habitat is. So it helps to kind of supplement that with a more mechanistic basis. And with that, we have survival. So here, I was also looking at how does survival change as a function of size and habitat use within the seascape, right? And for this, we had two basic hypotheses. There's a lot I'm sweeping under the rug here, a lot of covariates that we were considering. But the first is that the larger the animal is, the more likely it is to survive, the higher the probability of survival in any given habitat. The second was a function of habitat. We were thinking that the higher the structural complexity, so seagrass has highest, salt marsh intermediate, sand obviously has none, would confer corresponding probabilities of survival. Highest survival where you had the most refuge, lowest survival where you had almost no refuge, right? So that was the thinking. Getting into my methods, I focused primarily on the York River, which is a major tributary within the lower western portion of the Chesapeake Bay. And here for abundance, I used a stratified random sampling design where we stratified the river to kind of get at some of the spatial processes that we might not be accurately taking into account by just looking at some of these environmental covariates. And we were sampling at seagrass meadows, which are going to be in green, salt marsh edge habitat, which is SME, that's that brown dot, and then sand is that beige color. Now, one thing I want to point out is that seagrass in the York River is only downriver. It's not that I'm ignoring seagrass that are in the other two strata, it's that it's just not there. And so what we did was we went and repeatedly sampled from August to October of 2021. We collected about 120 samples, and this was done at biweekly intervals. For survival, I couldn't get it quite this many samples or this many sites, just for logistic reasons. And so here we were sampling from April to October of 2021. This time I took 848 samples. I actually collected over 1,000, but I had to throw out a bunch of them. So this may be a little bit of overkill. And we also did this at biweekly intervals to try to get, this was essentially the shortest interval that we could possibly get at that was logistically feasible given all of our other constraints. I spent a lot of time on a boat in 2021. In terms of how we actually collected these data, we used different gear for each different habitat, and we made sure to correct that with a bunch of efficiency testing to make sure that we were comparing apples to apples and not apples to oranges. For the marsh, essentially here we used something called a flume net. This took a lot of trial and error, but essentially we set up permanent traps where essentially when we weren't sampling, we buried these nets in the ground, and so animals could move across the marsh surface unencumbered. When we wanted to sample, we'd go out at high tide, we'd rapidly erect these net walls where the animals were basically staying in the salt marsh when it was inundated, right? And then we put in this cod end at the shoreline surface, right? Or the shoreline facing end of that column, right? And the thinking was that animals want to stay inundated. And so as the tide went out, animals would go from the marsh surface into the cod end as they were trying to stay inundated in that water. So at low tide, I would just walk up, hoist that bad boy right out, empty it on the boat, count and measure it. And then, of course, for good measure, I would walk through the marsh surface without breaking any grass, harder than you would expect, just to make sure that there were no animals that were like hanging out on the marsh or in the net or near a base of a chute or something of that nature. For the actual seagrass meadows, any of you guys who work in seagrass might know this glorified vacuum cleaner, the Venturi suction pump. Essentially what we do is we randomly within a seagrass meadow throw this mesh cylinder in a kind of a collapsed ring shape into the seagrass meadow. It's got a lead rimmed line at the bottom of the cylinder that gets weighted to the bottom. There's floats on the top to create basically a sealed column of water. And then I'd get in that with this suction pump, and I would essentially suction for 6 minutes. This is a great reminder of my master's work because I basically did the same thing with mollusks. Tom, turns out crabs are a heck of a lot harder to study than things that can't run away from you. In terms of sand, this was pretty basic. Most of you guys have probably been familiar with a seine net for some time. Essentially what we were trying to get at is that really shallow water unstructured habitat, because there's a lot of evidence that juvenile blue crabs are prioritizing shallow water where those larger fish can't actually access them. And so essentially what we would do is we would take the seine net, we'd start on one end of the shoreline and do this counterclockwise sweep, pull everything up, and then essentially count and measure it. So pretty straightforward. In terms of survival, I use something called tethering, which is a measure of analyzing emphasis on relative survival by essentially restraining these different prey items, in this case, juvenile blue crabs, within a given habitat. To give you an idea of what the schematic looks like, just like this. You essentially spend hours and hours in the lab up until 10 o'clock at night, super gluing fishing line to the backs of these guys' carapace with, and then putting a little bit of tape on there for good measure. To give you an idea, that's what it looks like. And then you stake these guys down in these different habitats. And the thinking goes that all else equal, animals that you get, and then you leave them for 24 hours and come back. So all else equal, habitats that confer higher survival, you get more crabs back on the line after that end of that 24-hour period. There are a lot of some assumptions with that. We had to do a lot of testing to make sure that this method is robust. I'm happy to take questions on that afterwards. And again, the whole setup looks something like this. We also wanted to keep a little bit of space between the pipe that we use to find the tether after that 24-hour period and the animal itself, just because a lot of predators are attracted to some kinds of these structures. And so this extra little bit of leash is essentially to correct for that, to make sure that they're actually, they're not being attracted by that pipe. In terms of the statistical design that I use, the first is a multivariate negative binomial model, so a count-based model. I know this equation looks pretty intimidating, so we're going to break it up step by step. Essentially, our response variable, instead of a vector, we actually regressed our covariates on a matrix, and this is going to be the response or the abundances of small, medium, and large-sized juveniles at any given site at any given time. We're then relating that using our log link function to these different environmental covariates. The ones I want to focus on today are going to be those different habitats that I talked about earlier. Then we were also interested in including a random effect. The thinking was that we were repeatedly measuring the same sites over and over and over again. Well, what if there were differences in current? What if there were differences in boater traffic? There were a lot of variables that we couldn't necessarily account for that we needed to. And so this random effect essentially encompassed some of those idiosyncratic characteristics in those sites that we were repeatedly measuring. The other thing that was obvious was, and that's our random effect, yeah. The other thing that was obvious was that today's big crabs are yesterday's medium-sized crabs, or the day before that small crabs. If you're thinking they might be related, you're darn right. And so what we actually had here was that for each size class, we included this covariance structure that allowed us to get at some of these dependencies, again, so that we could make better inference on those habitat variables. All of this other stuff are things that we want to account for so that we can get the best evidence possible about which habitats are the most important for which size classes. In terms of our survival model, and then of course, I'm getting a little ahead of myself. In terms of the different gears, we had gear efficiency testing, really thorough gear efficiency testing, and also differences in the areas that these gears encompass. So slight differences, a meter, 1.68 meters, 1 meter squared, 9 meters squared, et cetera. And so these other terms are essentially accounting for those differences in the gear that we used and the different areas swept. Again, everything to account for to make sure that our inferences are as robust as I could possibly think to make them. In terms of survival, here we just used a logistic regression design. This should be familiar to a lot of folks who have worked with binary data. And here our response variable are whether we got a live crab or a dead crab. Fun fact, when I used to give this talk, I used to label it 1 crab, 2 crab, dead little blue crab, because we were using abundance, counting abundance, and then looking at survival. But essentially, we were also regressing the probability of whether we got a 0 or a 1, live or dead crab, as a function of these different environmental covariates, and now looking at size as well. The reason we didn't look at size in the previous model is because size was basically implicit in those different groupings for those different size classes. And then again, we have our site-specific random effect for the same reasons that I mentioned previously. But here we also had a site trip-specific random effect. The reason is that we put out multiple tethers at each site at any given time. Well, what if a school of drum come by and just eat everything in those tethers, right? There's going to be within site trip dependencies that we needed to be able to account for. And so that kind of allows us to get at that part of the, basically account for some of those latent variables that we really couldn't think of any other way of accounting for. Again, to make sure that our inferences on these variables of interest, size and habitat, are as robust as we can make them. Getting into our results. Here, so I am a pretty hardcore Bayesian statistician, so I will usually refer to my effect sizes as posterior distributions. Essentially, these are going to be essentially the effects or the average density of these different animals or given size classes in these different habitats. Beige is going to be sand. Green is going to be seagrass, brown is going to be salt marsh. One thing I want you to pay attention to is that the scales are a little bit different. Salt marsh and seagrass start from 0 and go to 30, basically 30 animals per meter squared. Sand goes from 0 to 2. If I had this all on the same scale, sand would be a needle. And so I want this up there so that you can see it, but essentially I want you to ignore the column of sand for now. What you see here is that for these small juvenile animals, seagrass is obviously the big winner. There are a lot more animals, juveniles, in seagrass meadows relative to the animals that we found in salt marsh edge. Although surprisingly, we still found a good number of these animals in salt marsh. I have some ideas for why that might be. But as you move from medium and large sized crabs, you begin to see the shift, where increasingly animals are leaving seagrass meadows, or they're dying, either way, and you see these increases in marsh availability. When we look at this at an even more granular level, I'm actually redoing these analyses now, and you look at this at finer and finer resolutions of size class, these actually get way stronger. And so what we begin to see is that these animals are shifting to salt marsh habitat. And even at that 31 to 60 millimeter size class, they appear to be staying there. Yeah, there is a little bit of a decrease in the average expected abundance between medium and large sized animals, but that's almost certainly because of mortality. These guys are also getting eaten, right? And they're clearly not moving into seagrass meadows or into to sand habitat. However, again, abundance is just a correlative measure. And the first thing you learn in statistics is correlation cannot tell you anything about cause and effect. So we need to look at survival to provide a mechanistic basis for why this might be. And so when we look at our probability of survival as deduced from our logistic regression design, essentially here, these are going to be, again, posterior distributions, and the dot will correspond to the average expected probability of survival, and these error bars are going to essentially be the 80% credible interval. So we expect 80%, or the outcome is going to be, 80% of the time, the outcome will be, the true effect will be between these two intervals. And here in Downriver, you see essentially what we were expecting. Seagrass is by far and away the big winner in terms of the survival that it conferred for juveniles, regardless of size, followed by salt marsh and then unstructured sand. One thing that I did not expect is to see really high levels of survival in salt marsh in the mid-river and the upriver areas. That flummoxed me. And I think the best I can get at is there's a couple of caveats here, but the biggest answer is that in the estuarine turbidity maximum, again, that's going to be the area where there's a huge amount of food, tons of polychaetes, tons of amphipods, tons of bivalves. And so if you're one of many more things on the menu, odds are there might not be as much predation pressure on you, which might be giving us an indication as to why they're surviving so much better there. In terms of the size, I kind of got ahead of myself there. In terms of the size, essentially this was followed expectations. The larger the animal, essentially the higher the probability of survival. And so again, if you're a hungry red drum, you're eating a larger and larger blue crab, essentially you're going to live to fight another day. Emphasis on fight. Now, in terms of putting it all together, kind of putting the context together between the abundance and the survival, essentially here is our new revised paradigm for where these guys are landing in the different habitats as a function of size. And the survival, as well as leveraging some information that we had on growth, really begins to help us understand why these animals are making these decisions to shift to habitats at which size classes, right? So here, what you have is essentially the probability of survival as a function of tethering, so there's some caveats there, as a function of size on the x-axis, and then the green, brown, and beige lines correspond to the probability of survival in seagrass, salt marsh, and sand, respectively. And remember that salt marsh probability of survival changes depending on where you look at it, so there's a little bit of some wiggle room there. I want you to focus on this black line that goes across the graph. These are essentially equivalent probabilities of survival as a function of size. Notice that when you're a tiny little blue crab, you've got about a 60% chance of surviving in seagrass. But notice that the larger size classes get the same probability of survival increasingly in these salt marsh and even in the unstructured bottom habitat. This is important because remember, there's more food over there, particularly near the estuarine turbidity maximum. So essentially, if you can get the same probability of survival, but you get more food if you switch, well, there's now an economic incentive for these animals to be shifting to those different habitats. Economic from the perspective of crab, of course. And so this provides a mechanistic basis for why these animals are making those shifts at which size classes. Putting it all together now, essentially what we have is a seascape where these animals as post-larvae are emigrating, reinvading into these estuaries, and the first habitat they find is usually going to be seagrass meadows, and they're going to be strongly incentivized to settle there where they will rapidly metamorphosize. One of the things that we found was that there were still some animals that were winding up in salt marshes through idiosyncrasies of the currents and whether there's, there probably is some density dependence in there as well. But a fraction of these guys are not going to make it into the seagrass meadows, and they're actually going to land in the next best habitat they can find, which is salt marsh habitat. However, this picture begins to change as you see larger and larger animals, where the economic incentives for these animals to switch to these habitats change and becomes more advantageous for these guys to move to salt marsh habitat as they get larger because they get the same probability of survival, but more food availability. And my research indicated that these guys tend to stay there through that large juvenile phase. And even if you look at trawl data, you typically find high concentrations, particularly of adult males, in these same general areas near the estuarine turbidity maximum. And essentially what they're doing is they're migrating into that unstructured bottom near the estuarine turbidity maximum and just exploiting all of the food that they can eat over there. Now, taking this story and looking at it graphically, I know this is a lot. What I want you to focus on here is essentially this is all of the potential pathways an organism, this blue crab, can take as it moves to each cycle of its life within that juvenile phase, right? The research that we undertook essentially indicates that this is the dominant pathway. Now, this is important because let's reintroduce our people that I talked about at the very beginning of this talk, right? Essentially, what happens if we begin to implement these anthropogenic stressors on these different habitats? Say that we get rid of seagrass meadows, like what's looking like is happening in the Chesapeake Bay. Well, essentially, you've imposed a bottleneck. Now, the dominant habitat for these tiny blue crabs is taken away. And so you're going to have severe reductions in the population dynamics or the carrying capacity of the system if you begin to lose the seagrass meadow. However, say that we bring back seagrass meadows and we prioritize seagrass meadows at the expense of marsh habitat. Well, essentially, all we've done is shift the bottleneck over. We're still not going to have great productivity in terms of the number of animals that are being produced that will recruit to adult populations. I mean, you know, there's going to be some amount of transfer between seagrass meadows to those adult populations. They'll just remain there. But if I was a betting man, I would say that that amount of productivity isn't anywhere near what we would have if we had healthy, robust ecosystems or healthy, robust habitats that are used by every segment of this animal as it moves through its life cycle. And that's kind of the big takeaway. I don't really have text slides for my conclusions. This is it. We can't focus on one habitat. If we focus on only one nursery habitat that we know is important for an animal at the expense of others that are in their life cycle, we essentially are going to just shift bottlenecks over and we don't reap the benefits in terms of fisheries productivity. We need to identify every habitat that these organisms use and make sure that we prioritize and conserve them all. And with that, thank you so much for listening to me yap for 42 minutes now. I really appreciate it. I want to quickly acknowledge my advisors, Grace Chu and Rob Lipschus, my amazing committee over at the Virginia Institute of Marine Science, and my mentor at the National Marine Fisheries Service, Dan Hennon. I also want to thank everyone in my lab, including my amazing technicians and our lab students, my funding sources, and coffee, especially at the end. Oh boy. Coffee was definitely acknowledged in my dissertation. And then finally, all of my animations came from the University of Maryland Integrated Application Network. If you guys are trying to talk about your science and like using animations, that website is the one to go to. It is fantastic. And with that, I will say, oh, and of course, Gerald. Gerald was really helpful. With that, I will say thank you so much for listening to me talk and hearing all of my dad jokes. I love a captive audience.